Foods versus drugs. How did people ever begin to figure out what they could eat? And how did they figure out what were drugs? In 1996, Daniel Moerman published a study on the Native American uses of plants for drugs and food. He compiled a computerized database on Native American plant use, including medicinal, food, dye, and fiber use. He included 44,775 items from 291 tribes, both historic and current, and 3,895 species. He compared his data with the flora of North America. He asked, how did the numbers and variety of plants used by the American Indians for food or for medicine compare with all the plants that grow in North America, that is, with the North American flora? If use is random, then he expected to find more food or drug plants from large plant families and fewer from small plant families. And he performed a regression residual analysis to check his figures. What he found was that some plant families have many more or many fewer species used as food or used medicinally than you would expect from random distribution. So the American Indians used 2,564 out of the 21,641 available species in the flora, or 11.8% for medicines. And the top 10 families with more medicinal uses than expected, given the size of the family, included the aster family, parsley family, heath, rose, buttercup, pine family, the willow, honeysuckle, mint, and oak family. Among families avoided for medicinal use are those that produce the bulk of human food, that is, the grass family and the bean family. The bottom 10 families with less than expected drug use include the orchid, mustard, bellflower, borage, pinks, madder, figwort, legume, sedge, and grass families. However, some families provide both medicines and foods, and in particular the rose family and the tomato family. So the top 10 families producing more food than you expect, given the size of their family in the flora of North America, include the lily, rose, heath, parsley, goosefoot, oak, gooseberry, cactus, pine, and knotweed families. Whereas the bottom 10 families with fewer food plants than expected include the cotton, bellflower, aster, phlox, orchid, pinks, spurge, madder, figwort, and sedge families. Other researchers, for example Timothy John, hypothesized that in selecting plants for food, people found drugs. But based on this research, Moerman argues that food and drug plants are different spheres of knowledge. So what is a food and what is a drug, according to the U.S. Food, Drug, and Cosmetics Act? Well, a food are things used for food. You can observe them being eaten. Whereas drugs are things that are not food, defined in terms of intent, not behavior. In Moerman's database, 1,625 species are used as food. Now, other primates and a few selected vertebrates use anywhere from 6 to 100 species for food, with an average per species of 43. So when you look at individual human groups, not in the aggregate as I first stated up above, we see an average of 58.5 plant foods per human group. So the human variety in food is comparable to the variety eaten by other primates. But the human use of medicines is clearly very different from other primates. Individual primate species use 1 to 2 species of plants. And you might wonder how we study this. Researchers follow primates in the wild, and they observe them often through binoculars and watch what they eat and how they eat it. and then they collect the feces that drop to the floor. So many of these primates are way up high in the tree canopy. And they can analyze the feces to see if the primate was ill or whether they were pregnant. And what they've observed is that they sometimes pick out species of leaves that they don't normally eat, except when they are ill, or they might eat it in a way they wouldn't normally eat, such as rolling up the leaf to eat it. Humans use 2,564 species of vascular plants. 642 species are used only as food. 1,581 species are used only as drugs. And 983 species are used both as foods and drugs. So, humans use far more species as drugs than as food, whereas the opposite is true of other primates. They use far more species as food than they do drugs. Moermann argues that if Johns is correct, that is, that in selecting food people found drugs, then we would have more plants used as both food and drugs. Human food is always meaningful. Human medicine is meaningful. Human food and drug plants have very complex origins. Food provides energy and protein and complex secondary chemicals that promote health. Drugs are similar, but often in dramatically different proportions. Altogether, 21% of species produce proportionally large shares of both food and drugs in Moermann's study, comprising 37% of drug plants and 49% of food plants. These include especially 4 families. The rose family, which has 836 different North American species. Among American Indians, 23 of these species were used for food only, 56 for drugs only, and 77 for both. In the lily family, which has 509 species in the North American flora, 55 were used by the Indians for food only, 26 for drugs only, and 39 for both. And in the mint family, which has 469 species in the flora, 7 were used for food only, 38 for drug only, and 34 for both. And finally, in the tomato family, which has 202 species native in North America, 10 were used for food only, 14 for drug only, and 20 for both. So, at the level of family and species, many food and drug plants are the same. But often, when the same species is used both as food and drug, different parts of the plant are used for food versus for drugs. So 58% of fruit uses are for food, but only 8% for medicine. Whereas 5% of whole plants are used for food, but 22% for medicine. 13% of roots for food, but twice as many, 27% for medicine. There are also substantial differences in the plant part used among different families. Food, we tend to use either the plant tops or the fruits in 82% of 28 families. Whereas in drugs, we tend to use either the roots or the plant tops in 63% for 28 families. Food and drug plants also tend to have different growth habits. Food tends to come from a shrub or a tree. Drugs come from shrubs, trees, lianas, semi-shrubs, and vines. Whereas grass, fern, and grass-like plants produce relatively few foods or drugs. Drug plants tend to be perennial, as do plants used for both food and drugs, whereas food plants tend to be annual. Drugs are used both internally and externally. Foods are eaten, thus they are used internally. Medicinal taxa used internally are also likely to be used externally. To Moermann, all of this taken together suggests that food knowledge is distinct from drug knowledge, but overlapping. He says that while people certainly learned of the medicinal uses of foods, they also developed a knowledge of medicines independently, using different analytical techniques. Moermann infers that our food knowledge is older, whereas medicinal knowledge has accumulated just since we became the genus Homo, or perhaps even just since we became Homo sapiens. Timothy Johns, in one of his publications on plant constituents and the nutrition and health of indigenous peoples, talks about how indigenous societies define themselves in relation to people and to their resources. So when modernization disrupts local ecology, it also disrupts their sociocultural system. And since health and nutrition have an ecological basis, modernization also has negative impacts on native health and nutrition. Johns argues for the need for traditional societies to control their traditional lands, that is, their resources. The traditional people need the resources for their food and health, and they are usually good long-term caretakers. He provides examples of what happens when the integrity of traditional systems is destroyed. All humans need to derive essential nutrients from their diet. And depending on the local environment and plants, local populations may develop enzyme polymorphisms. That is, they may be able to digest plant foods that other people can't digest. Or they may like the taste, the strange or unpalatable taste that other people would not like. So loss of traditional food systems affects not just nutrition, but also health. The population may face increased exposure to toxins because they might have to eat famine foods, which, as you know, might have more allelochemicals in them. Or they don't know how to correctly process the new foods in the new system that they've got after the loss of their traditional food system. Or they may be exposed to environmental toxins. And the refined, industrialized diet may not be as healthy as the traditional diet. And we'll talk more about this on Friday. Some plant allelochemicals may promote disease, although others may prevent disease.